students, in first part of this chapter, osmosis, absorption by root, we had studied the adaptation of root, the conditions in which it absorbs water and minerals from the soil. We had studied two modes of absorption of water by the plant cells or root, that is the diffusion and imbibition. Now, this chapter is mainly based upon the third phenomenon, third mode of absorption, that is called osmosis. Now, so the next part is osmosis. Third part is osmosis. Now, students, we have studied that in diffusion, the movement of solute as well as solvent particles take place from region of their higher concentration to the region of their lower concentration. When the two are kept in direct contact, means in diffusion, the bidirectional movement can take place, and it will stop only when the concentration become uniform. Now here in osmosis, the movement of mainly solvent, that is movement of solvent, mainly water, takes place when the two solutions of different concentration are separated by a semi-permeable membrane. Now students, here the term is used as semi-permeable, means the membrane which will allow movement of solvent, mainly water, and prevent the movement of solute particles. So what is osmosis? The movement of solvent, mainly water, from region of its higher concentration. Now student, we see that is movement of its higher concentration. Here the its word is used for, that is water. So movement of water from region of its higher concentration, and if the water is in higher concentration, that will be less concentrated solution, that will be also known as hypotonic solution, which will later on describe in detail. Hypotonic solution to the region of its lower concentration. That is, if the water is in a lower concentration, then it means that it will be more concentrated solution, it will have higher concentration of solute. So lower concentration, that is more concentrated solution, or this is also termed as hypertonic solution, across a semi-permeable membrane. Membrane is called osmosis. So a student, here some terms are required to be written clearly. The two solutions, that is water moves from region of its higher concentration. Clearly it is written here, means the water is in higher concentration with respect to the solute, means there is less solute, hence called a less concentrated solution. We can also use the term dilute solution. And to the region of its lower concentration, means less solvent, less water, hence more solute, so termed as more concentrated solution, also termed as hypertonic solution, across a semi-permeable membrane. Here we remember, that is the two solution must be separated by a semi-permeable membrane. Then this phenomenon is known as osmosis. So clearly, a student, you see that semi-permeable membrane semi-permeable membrane allows movement of solvent that is water, but prevents movement of movement of solute. So this is the semi-permeable membrane. Our cell membrane behaves like a semi-permeable membrane. Cell membrane is also termed as selectively permeable membrane. So in semi-permeable membrane there are few examples, that is cell membrane also termed as plasma membrane, cell membrane, egg membrane. Now cellophane paper, goat's bladder, further whisking bag, that will be studied later on, whisking bag, etc. So these are the different examples of semi-permeable membrane. So in this case you must remember, that is, what are the conditions required for the osmosis to take place? First, the two solutions must be having different solute concentration, and second, the two solutions must be separated by a semi-permeable membrane. So if we write the conditions for osmosis to take place, conditions for osmosis. First, there should be two solutions having different solute concentration. First point. And second, the two solutions will not be in direct contact, that is, the solutions should be separated by a semi-permeable membrane. So just you see, a student, that in these two conditions, if any one of the conditions will be unavailable, the osmosis will not take place. So that concept can be used while arranging the experimental setup and arranging the control setup for that experiment. We will require the two conditions, that is, in control setup, if we'll change any one of the two conditions, like if we take two solutions of same concentration separated by semi-permeable membrane, there will be no net movement of water, there will be no change in concentration. And if the two solutions of different concentrations, if separated by, in place of semi-permeable membrane, by an impermeable membrane like rubber sheet, plastic film, or a freely permeable membrane like muslin cloths, which will have large pores through which solute particles can also move. As a result, in the two conditions, osmosis will not take place. So while arranging the control experiment, we can change any of the two conditions. Clear, student? So we have discussed just osmosis, its conditions, and the available conditions or factors responsible for osmosis. Now we will clear it with the help of an experiment. Now in ICSC, experiment based questions are asked in which the diagrammatic setup will be given. There are point wise questions because descriptive questions are not asked in ICSE. If the question will be of 5 marks, there will be 5 parts in questions and each part will carry 1 marks. And there is a definite pattern of question, like if the diagram is given, question will be asked, what is the aim of the experiment? Then you will write the phenomenon which is being studied. Like suppose we arrange the experiment for osmosis, then aim of the experiment will be to show the process of osmosis. Sometimes it is directly asked, name the phenomenon being observed. It will write like that. Then the question will be asked, what will we observe after some times? Then we will write its observation. In the third part, the reason may be asked. Give reason for your above observation. In the fourth part, you can have to define the process as well as you can have some related questions to it. So there are many questions asked. So suppose we are going to study experiment on it. Experiment on it. Suppose we take experiment number 1. So first of all, we will write aim of the experiment to demonstrate the process of osmosis. Now, how will you arrange the process diagram, experimental setup? First of all, we take a long stemmed inverted funnel, or we take a long stemmed funnel. At the end of the funnel, we tie semi-permeable membrane firmly, like it may be cellophane paper, the most easily available. Now, then we fill some concentrated sugar solution in it. Now, suppose we mark here, this is any semi-permeable membrane, like suppose we take cellophane paper, any semi-permeable membrane. Now, we fill it with suppose sugar solution. And then we mark the initial level of solution in the funnel. Then this tube is immersed by inverting in a beaker or a trough containing pure water. Now suppose here is pure water. So now you can see there are two solutions. One, the sugar solution inside the funnel, and outside it, that is in the beaker, there is water. So there is difference in concentration of solute. Then after that we will have to observe it. What will you observe? So you will have to write here observation. After some times the level of solution in funnel rises. Means we will observe gradually the rise in level of the solution. Suppose at that time we mark this level as the final level. So this is your observation. Now the next part is asked that give reason for your observation, above observation. Now we see that inside the funnel there is sugar solution that is more concentrated. In the beaker there is water, pure water, so this will be less concentrated. So the water from outside, where it is in higher concentration than the solution inside the sugar. So water from the beaker moves into the solution inside the funnel across this semi-permeable membrane. So here we write that is water in beaker is hypotonic hypotonic, that is less concentrated than sugar solution inside the funnel. So water from beaker moves into the sugar solution across semi-permeable membrane by osmosis. That is, suppose the question is asked, name the phenomenon studied in the diagram. We will write the process of osmosis, the phenomenon of osmosis. So this is the region. Now one more question is asked, as just I have discussed earlier explaining the semi-permeable membrane, that or the condition of osmosis. Suppose here we need to write about its control. Sometimes question is asked that what will be the control setup for it. Now in the question it will not be given, but we will have to make the arrangement, or we will have to write the conditions. So what will be the condition required? So we have discussed there may be two cases. Any one of them can be written, but just I am explaining the two. That is, we can use same solution, that is sugar solution or water in beaker and funnel. That will be the same concentration. So no osmosis, because there is a same concentration of solution. Or in place of semi-permeable membrane, we can use impermeable membrane. Impermeable membrane. It may be plastic or rubber sheet, which impermeable membrane we can use impermeable membrane, or a freely permeable membrane, membrane like muslin cloth, which will have large size of pores through which solute particles will also pass. So if we will use any one of the two conditions, there will be no osmosis to take place. So the question will be asked like this. Just again I am explaining you. In the exam the diagram will be given, and you will have to, after studying the diagram, you will have to answer the questions. Suppose the first question is, what is the aim of the experiment? You will write like this. In second part it will be asked, what is the observation made by you? So this will be the observation. And then the question is asked, give the reason for the above observation. You will write like this. It can further be asked, what will be the control setup for it? It can be written, or it can be asked to define the process. So experiment based questions are asked on plant physiology. Now a student, in this type of questions there may be some modifications. Diagrammatic questions, there may be some modifications, like you see. Suppose In this part, as we have studied sugar solution here taken and here water, we can have sugar solution at both places, but there should be different concentration. Like suppose we take 60% sugar solution inside and 20% sugar solution outside. Again you will have the same type of answer, because just we have changed the concentration of the sugar solution. One thing. Next, sometimes the question is asked like that suppose outside will take more concentrated solution. Like you see, suppose as a normal condition, suppose just I am changing the question. Now suppose this was the initial level. Now the sugar solution inside the funnel was 20% and outside the solution, that is in beaker, it is 60%. In that case, there is more water concentration inside the beaker, inside the funnel than in the beaker. As a result, water from the beaker funnel moves outward. That is, in this case, solution inside the funnel is hypotonic, less concentrated, more dilute than the outside. Hence, water from the solution inside the funnel moves outward, that is into the solution, into the beaker. So this will be the final level. So what happens here? The level of solution decreases. Means this time again, depending upon the concentration of solution, the direction of movement of water has changed. Clear, student? Now, on the basis of this direction, there may be two types. First, endosmosis. As the name suggests, endo means inward, inside. So this is inward movement of water across semi-permeable membrane when a cell is kept in hypotonic solution. That is, cell is surrounded by less concentrated solution, that is more dilute solution, is called endosmosis. Now, if the endosmosis takes place, then the volume of the chemical substances, water inside the cell will increase. So it causes swelling of the cell and it turns turgid. That is, the cell will increase in size. But if there is exosmosis, in that case, as the name suggests, outward movement of water takes place. So the outward movement of water across semi-permeable membrane when a cell is kept in a hypertonic solution means more concentrated solution. Now you see outward movement of water across semi-permeable membrane when a cell is kept in hypertonic solution, that is more concentrated solution. So this is the outward movement of water. In that case, what will happen? The cell sap, the cell content will decrease as the water has moved outward, so it causes shrinkage of cell and it turns flaccid. So this is that is according to the direction of movement of water. Osmosis may be endosmosis or exosmosis. But whenever the question is asked related to the diagram, you will not define these two terms, but you will only define osmosis, because the process is osmosis. You need not require to write endosmosis or exosmosis. Whenever it will be asked only, then you can write. Clear, student? So this is about the experiments related to osmosis. Students, after explaining an experiment, its conditions, the basic requirement, the basic questions asked to it, we can have some more experimental setup which can be given in our questions. Now you see, in some questions we have seen, that is in some model test papers, this type of diagram will be given. Suppose this is a long U tube, and in the middle of it we will have semi-permeable membrane. That may be anything, just we have discussed. And then in the two limbs we will place two different types of solutions. Like suppose in limb A we take 60% sugar solution, and in limb B suppose we take 20% sugar solution. Now this is the setup. So the question will be asked directly again like that, what is the aim of the experiment? In that case, we just recognize here, that is there are two different concentration of solutions, and there is a separation of semi-permeable membrane between them. It means that it is related to osmosis. So the aim of the experiment is to demonstrate the process of osmosis. Now then what will be your observation? You will observe that the level of solution in the limb A will gradually increase, and the level of solution in the funnel in the arm B or limb B will gradually come down. Now what is the reason? The solution on limb A B is less concentrated, that is has higher concentration of water than the solution in limb A. So by the process of osmosis, water moves from side B to side A. As a result, we will see that the level of solution in limb A will increase and in limb B it will decrease. Clear, student? Now other questions like control and other things can be explained in the similar way as the first experiment. Now suppose there is second experimental setup. In this experimental setup, suppose just we have a vessel in which in the middle we will have a wall of, suppose like this, we have this type of beaker and we take this. So this is the semi-permeable membrane, wall of semi-permeable membrane. Semi-permeable membrane. And again similar to this question, we will have two different solutions on side A, on side B. here is the 60% sugar solution, and on this side there is 20% sugar solution. Now, what we'll observe after some times, again similar to this one, the solution on side B is less concentrated, that is higher concentration of water. So across this membrane water moves by the process of osmosis. As a result, we will observe that level of solution will rise up on side A and it will come down on side B due to the process of osmosis. Now, if the reason is asked, again you can explain in the similar way. That is, there is higher concentration of water in the solution on side B. Therefore, water from solution on side B moves into the solution on side A by the process of osmosis. So, the level of solution inside BA will rise, whereas it will come down on the side B. Means, a student, in the examination we can have any shape of the experiment, but how will it be recognized? Just we will observe there should be two solutions of different concentration and there must be a separation of semi-permeable membrane. So wherever this condition is available, we will answer for the process of osmosis. Clear, student? Now take one more example. One more example can be taken here. Now, suppose in another example, suppose third, suppose we have a narrow tube at the end of which we tie a whisking bag like this. Again in this is the whisking bag, a kind of semi-permeable membrane. Here we take sugar solution. Now this is initial level of solution, and in the beaker we take water, or we can have less concentrated solution. After some times we will observe that the level of solution in the tube will rise. Means this is the final level of solution. Now this is again demonstrating the process of osmosis. Now a student, in the board examination, on the basis of this some question was asked, that is, compare the different parts with plant. So first of all we see that this phenomenon takes place in the root hair cells, because root hair cells absorb water from the soil. So question will be asked, in which part of the plant does the phenomenon take place? So we write it takes place in the root hair, or we can write root hair cell. Then the question is asked to compare these structures. So first of all we will write here solution in the funnel. It is similar to the solution inside the root hair cell. So it will be cell sap in root hair. Next, the water surrounding it, across which the osmosis is taking place. So the water in the vessel or in the beaker, it will be similar to the soil water, because the root hair cell is surrounded by the soil water and which moves across the cell membrane into the root hair cell. Third, this whisking bag. Now the whisking bag is semi-permeable membrane. So in the plant root hair, where this phenomenon is taking place, is the cell membrane in root hair. cell membrane in the root here. Clear, student? So this type of question is asked to compare with the plant parts. And just we have studied, a student, that in osmosis you can have various setup, various experimental diagrams, and just by comparing the concentration of solution and the presence of semi-permeable membrane separating the two, we can answer about the osmosis. So it will become easier if you learn, if you will recall, if you will remember only these two conditions required for osmosis, you can answer any type of diagrammatic question. Clear, student? In the next part, we will study the different terms related to osmosis and other things. It will become quite clear to you. Okay, student? Thank you.